Professor SNAB, where we make A-level biology as simple as a cell. So today we're understanding and looking at a core practical in which we'll investigate plant mineral deficiencies. So the aim of this investigation is to look at the effect of mineral iron deficiency on plant growth by growing plants in nutrient solutions lacking specific ions. So plants require inorganic ions for growth. So if a specific mineral ion is absent, plants show characteristic deficiency symptoms. And so by removing one mineral ion at a time, the role of that ion can be determined. So first, you need to grow genetically similar seedlings. So one example is barley in hydroponic nutrient solutions. Hydroponic nutrient solutions are water-based mixtures of essential minerals. Use a complete nutrient solution. So this is a control variable, and also use solutions each lacking one specific mineral ion. So you need a solution that lacks nitrate, you need a solution that lacks magnesium, and you also need a solution that lacks calcium. Ensure that all variables are controlled, such as light intensity, temperature, volume of solution, and pH. Grow the plants for a fixed period and make observations and record the plant height, leaf color, and root development. We'll look into the analysis side of this practical. So you need to take note of the fact that the independent variable is the type of mineral ion missing from the nutrient solution. The dependent variables are the plant height, plant biomass, leaf color, and root development. And then finally, the control variables in this practical are light intensity, temperature, time, and plant species, and also age. So what kind of results will you be expecting from this practical? So the deficiency of nitrate leads to stunted growth and yellow, older leaves. With the deficiency of magnesium, you'll observe chlorosis of older leaves. With deficiency of calcium, you'll observe poor root growth and distorted leaves. And finally, in the case of a complete solution, you will observe healthy growth of the plant. So what are some evaluation points that you can consider for this practical? So the limitations are the mineral ions may be stored in seeds initially, so the results you observe might not be of the highest quality. The visual assessment of colors can be subjective, so your results can vary from person to person. And finally, the sample size investigated is small, so it might not be the most accurate observations. So now to look at how we can improve our practicals. So use replicates, so repeat the practicals. You measure the dry mass rather than the height. You can also grow the plants for a longer time duration, so you can actually observe the results. And another one, this is quite an interesting one, is to use a colorimeter to quantify the leaf color, because leaf color is subjective. So colorimeter gives you the opportunity to quantify this data. So improving the quality of the data you collect. So what are some safety and ethical considerations you need to make for this practical? So you need to wear eye protection when handling nutrient solutions, and also ensure that you dispose the solutions in a responsible manner. Your lab tech will give you the guidance to do this. Do not go about to throw the solutions used down the sink, for example. That is irresponsible. So that is all from Professor SNAB today. Hope you found this video useful. If you have any questions, please let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share and subscribe.